we started the respiratory system. A good bit of the anatomy and the function of breathing we're going to cover in lab, or we did for half a week. What I want to concentrate on today is gas exchange. Oh, oops. Is gas exchange, or how oxygen and carbon dioxide are exchanged between the air and the blood. Is that too dark? So to begin with in this section, it refers to some gas laws, which I think I talked about last time, the partial pressure thing. There's Dalton's law that basically says that the pressure of a mixture of gases is simply the summation of all of the partial pressures of each gas. So for the gas that we're talking about that makes up air, nitrogen, oxygen, carbon dioxide, water vapor are the major players. There are other gases that are present, but they're all in very low quantities. So this table here is showing the percentage that the air we breathe is made up of nitrogen is about 78%. The partial pressure is 597.4. Oxygen is just a little bit below 21%. It's about 159 millimeters of mercury. Water in its vapor form, gaseous form, is about 0.04% of the air we breathe and represents about 3 millimeters of mercury. Carbon dioxide is 0.004%. And therefore is about 0.3 millimeters mercury worth of pressure in the air that we breathe. And then the others, just the various gases that fit into this scheme that we're not going to deal with individually because they're so rare. You know what I mean? They're such a small component of the gas we're breathing. We'll just lump them together into others. They equal 0.0006% total, which is about that's weird. 0.5 millimeters of mercury. That's got to be wrong. That must be like 0.05 or something like that. But the point is that all of these pressures add up to 760 millimeters of mercury pressure, which is air pressure at sea level. As I was talking about this last time, it occurred to me to ask about people that had, I had asked about Samsung phones, but after class somebody came up and showed that the latest Apple also has this. Phones are starting to have incorporated into them barometric sensors. So your phone has a GPS sensor, or well, radio, and it has a gyroscope sensor and various different kinds of sensors. It's kind of escaping me off the top of my head all the different sensors that are found in a phone, but some phones are starting to incorporate a barometric sensor, which is useful in terms of this because the pressure of air varies as we move up from sea level. And from that sensor, you can, if you have the correct app running, you can get an accurate reading of the pressure of the air right here. What I was interested in, and I was kind of hoping to pursue this, and I didn't get to between last class and this class, was to get a good sense of the difference in pressure between ground level outside in the quad, and up here on the 5th floor. I was kind of curious as to how much pressure changes just in that little bit. Jessica, was it you that showed? Yeah. So using Jessica's phone, we saw that we're like, I want to say 67 meters above sea level here. Which would be, let me think about this. Probably about 200 feet above sea level.
Yeah, we're at 226.
Yeah.
232.
Okay, so not bad math in my head, kind of figuring that. But so that's how far above sea level we actually are. But I also, and Jessica, I'll put you to the task after class, get an accurate reading of where we are on this floor versus where you're at the bottom. I'll just be curious how much change that is. But anyways, we can use that information here. And really, these numbers here, the partial pressures, aren't individually available exactly. We know what the total pressure is, and we know what the percentage of 597.4 here is just 78.6% times 760. And that's why I think this other one down the bottom is wrong, because if 0.004% times 760 millimeters of mercury gives us 0.3 millimeters of mercury pressure, then a smaller number, 0.0006 times that, should give us a much smaller number here. I wouldn't be surprised if it's like 0.005 or 0.05 or something like that, and they just messed up the decimal point there. But we don't really care about the others there. We're really just going to pay most attention to oxygen, carbon dioxide, and the nitrogen and water stuff comes up as we're thinking about this. But we're going to pay most attention to the oxygen and carbon dioxide because those are the ones that are important for how things work. And then the other thing was, what's the guy's name? The other law, Henry's law, which was about solubility of gases and liquids. Just that whatever the partial pressure is of a gas in an air medium is going to correspond directly to its how much of it is dissolved into a liquid medium. All that really means is that gases diffuse through air and liquid equally. So if you have an air phase and liquid phase next to each other, the gas, whichever of these gases it is just going to diffuse across between the two until it's equilibrium. It's at equilibrium across the entire space, which is important because we're thinking about oxygen or carbon dioxide in the air in our lungs and oxygen and carbon dioxide within our blood. And we can basically treat those two components or two compartments equally. The oxygen that's in your lungs will move into your blood easily, as if there's no difference between the air and the liquid media, and vice versa, the carbon dioxide will move. So we can just treat the air movement or the gas movement as unimpeded between the two because of what Henry's law basically says. Now, ventilation is really what we mean by breathing, and that's something that we're going to focus on for lab. But what that means for gas exchange is merely that getting the air in and out of the lungs is important. Now, for gas exchange, there's two things that we're going to look at. There's external respiration and internal respiration. External respiration is the movement of gases between the air in our lungs and the blood. Now, this figure that is taking a little bit of time to reload is showing just that. Oxygen in the air that we breathe is going to move into the blood, and then carbon dioxide from our blood is going to move back out into the air. Okay, here we go. So we see oxygen in the alveolus, meaning in the air that we're breathing in, moves across the alveolar wall and across the blood vessel endothelial wall and goes into the blood. Some of it goes directly to dissolve in the plasma, but the majority of it goes into the red blood cell. These two arrows are not really drawn to scale or something like that. The amount of oxygen that just dissolves into the plasma is less than a couple of percent. It's usually 1.5% is this number I usually see. And the other 98.5% of the oxygen that gets into the blood goes into the red blood cells. Now, there is a little bit of oxygen in the plasma, but we're actually going to kind of ignore that because it's such a small percentage and it doesn't do anything terribly interesting. It's just dissolved in the plasma. The stuff that goes into the red blood cell, on the other hand, does do something interesting, which we're already a bit familiar with. Oxygen combines with the iron and hemoglobin, and that's how it's carried in the red blood cell. Now, hemoglobin is abbreviated HB, and you can see there O2 plus HHB, because when oxygen's not bound to hemoglobin, something else will be. And the H in HHB is referring to hydrogen. Hydrogen ions, which really means just protons, will take up a position attached to the iron and hemoglobin in place of the oxygen. And then when oxygen comes in, it just supplants the hydrogen ion. So then we get HbO2, which means oxygenated hemoglobin or oxyhemoglobin. And then the hydrogen ion on the other side of that equation here, okay, right here. Now this arrow is backwards, which hopefully should be obvious because the oxygen's coming in, so the oxygen's going to react with hemoglobin. And we're going to get oxyhemoglobin, and then the hydrogen ion comes off of that. That's what oxygen does. It just comes into a red blood cell and attaches to hemoglobin. Okay. Not all of the hemoglobin is actually attached to hydrogen. Some of it is attached to carbon dioxide. Okay. So in this picture, they're showing carbon dioxide being released from hemoglobin to get into the air and lungs. And we'll get to the carbon dioxide side of things in a little bit. But really, some of the oxygen is going to attach to this hemoglobin in carbon dioxide's place. Now, what's happening with the carbon dioxide side of things is a little bit more complex, chemically speaking. And in this figure, they highlight the chemical reaction that takes place here. Okay. Now, what we're trying to do is to get carbon dioxide out of the blood and into the air. The majority of it is coming from a compound, which is really carbon dioxide and water together, okay, which is H2CO3. An enzyme called carbonic anhydrase catalyzes the breakdown of H2CO3 okay, into CO2 and H2O. Okay. Now let me go over here for a second and CO3 breaks down into H2O and CO2. And I can't do subscripts on this, but you can see what we're talking about. H2CO3 is called carbonic acid. And then obviously H2O is water. And CO2 is carbon, oops. dioxide. Okay. So carbonic acid is really just the compound that's made of a water molecule and a carbon dioxide molecule. Now, the enzyme that breaks these down, which was in that figure we just looked at, is called carbonic anhydrase. So the carbonic in this name is referring to carbonic acid. And then anhydrase is referring to an enzyme that removes the an, whoops, come on. The an means to remove, and then hydra or hyder is referring to water, as in the word hydrate. Okay, so to anhydrate something is to remove water from it. Now that sounds like a really weird word, and you would normally say dehydrate, but chemists have their different ways of naming things. So anhydrase is an enzyme that removes water from carbonic acid. That's what the name means. So if you remove H2O from H2CO3, what's left over is CO2. Okay. So it breaks carbonic acid down into water and carbon dioxide. That's what the enzyme's name is telling us it does. Okay. And that's what we see here. The carbonic anhydrase is broken down into water and carbon dioxide, and the carbon dioxide is then released and it moves into the gas, into the air in our lungs. Now, the carbonic acid is not just sitting around in our blood or our red blood cells. It's actually going to be in an equilibrium between H2CO3 and then the H plus ion, the hydrogen ion, and HCO3 minus ion, which is called bicarbonate. So to call something an acid really means that it can, I should say this, can sort of slip back and forth between forms where we have a proton, a hydrogen ion, and the rest of the molecule. Okay. Oops, I left out the H there. Now this HCO3 minus is called bicarbonate. The bicarbonate ion. And so carbonic acid, when it's in solution, occasionally a proton will come off, and a proton is a hydrogen ion. And what's left over is the bicarbonate ion. Okay. That's the way acids basically work in solution. They have the ability to lose a proton, which is going to therefore increase the pH of the solution. Okay. pH is just the concentration of hydrogen ions in the solution. So as an acid loses more and more of these things, these hydrogen ions, the pH becomes more acidic. And what's happening here is going to go back and forth until it's in equilibrium between the two. But if we're breaking down the carbonic acid to release the carbon dioxide, then more of the bicarbonate and hydrogen ions are going to form together to make the carbonic acid to get it back into equilibrium. And then we break that down to release the carbon dioxide. And we're going to be basically removing these things from the solution. Okay. Now, the hydrogen ion here basically is being released from hemoglobin when oxygen binds to it. Okay. It's not stated in this picture here just because it's getting confusing enough. But the bicarbonate actually is coming in from the plasma. Okay. Bicarbonate by itself works as a buffer. It helps to limit the change in pH of, in this case, the blood plasma. Okay. You're probably familiar with that reaction. You might not realize it, but if you've ever taken Tums or Rolaids or something like that, you've basically been doing that, but in your stomach. So Tums is calcium carbonate, and I think Rolaids is sodium bicarbonate. But whichever form it is, it has a buffer in it like this stuff that will de-acidify your stomach acid. And actually what happens there is exactly what we see here, that it forms carbonic acid, and then that can break down and you release CO2. And when that's coming out of your stomach, that's usually the gas that's coming up in a burp. Okay. But in the blood cell, this is happening here to release the carbon dioxide into the air in the alveolus. Okay. So this is basically what's going on in what's the word I'm looking for here? External respiration, what we've been talking about here. Now in the book, after they explain external respiration, the next thing they get into is internal respiration. And they have another picture here. This picture has tissue cells, which are just sort of some generic cell drawn, and a red blood cell in floating around in the plasma in the bloodstream. We see carbon dioxide moving from the tissue into the red blood cell and oxygen moving from the red blood cell back into the tissue. Okay. This picture and this picture are very similar, only that we have an alveolus on the left side of the picture and this an external respiration, and we have tissue on the left side of the picture and internal respiration. And then also carbon dioxide is moving left to right in the picture in internal respiration, and it's moving right to left for external respiration. And oxygen is moving left to right for external respiration, and it's moving right to left for internal respiration. So things are just running backwards. Okay? Internal respiration is just the exact opposite of external respiration. The external respiration picture has the chemical reactions that are taking place inside the red blood cell. The internal respiration does not, but it's essentially just the opposite of what we have here. So carbon dioxide is taken up, it combines with water, carbonic anhydrase puts those together to make carbonic acid, which will dissociate into hydrogen ions and bicarbonate. The hydrogen ions then replace the oxygen on hemoglobin. So for the external respiration picture, this part is actually backwards. The arrow is pointing the wrong way. And but if we're using this to talk about what is going on in internal respiration, the arrow is pointing the correct way if it was in this picture right here. Okay. The carbon dioxide that comes in is also going to attach to hemoglobin to some degree, and we get this carbaminohemoglobin, which is carbon dioxide attached to hemoglobin. Now I said in this picture that these arrows aren't really drawn to scale. Only about 1.5% of the oxygen dissolves in the plasma, and the rest of it goes into the red blood cell. In this picture, it's a little bit more suggestive that there's very little oxygen from the plasma and more coming from the red blood cells. Because again, it's just the reverse. So this is going to be 98.5% of the oxygen coming out of the red blood cell, and only 1.5% that's coming directly from the plasma. They don't show the different kind of weighted size, whatever you want to call, of the arrows here for carbon dioxide in either picture, this one or this one. The arrows look kind of similar. But in fact, the majority of the carbon dioxide is going to be tied up in the carbonic acid reaction. About 70% of the carbon dioxide goes into that. About 23% is going to be attached to hemoglobin, and the last 7% is in the plasma. Okay. So just for external respiration, about 98.5% of oxygen goes to the red blood cell, and about 1.5% goes to plasma. In internal respiration, 70% of CO2 goes into carbonic acid. Oops, 23%. Should continue with that. Goes to hemoglobin, and 7% CO2 goes to plasma. So this is just kind of how it breaks down. The book doesn't have that worked into those pictures, which are already kind of full of information as it is, but that's a breakdown of where things go. I'm saying oxygen for external respiration and carbon dioxide for internal respiration, just talking about what's going into the blood, okay? Oxygen is going from the air into the blood in external respiration. Carbon dioxide is going from the blood out in external respiration. Carbon dioxide is going from the tissue into the blood in internal respiration. Oxygen is going from the blood into the tissue for internal respiration. Okay. And so as oxygen comes out of the blood or carbon dioxide goes into the blood in the opposite, the percentages are the same. That's not changing, but I'm just highlighting where it's going into the blood, okay, just so we can sort of separate that out. Now, in talking about gas exchange, we're talking about external respiration and internal respiration, which is what's happening between the lungs and the blood and what's happening between the blood and tissues. In between that, the gas is going to be transported through the blood. And so this section we're just looking at here is dealing with both aspects of gas exchange. The next part is looking at the transport of gases, how the gases are moving through the blood. And this is what's happening between external respiration and internal respiration. So for oxygen, like I said earlier, it's being transported in the blood by hemoglobin. And here's a representation of the hemoglobin molecule. It has 4 subunits, each of which has a heme molecule in it with an iron ion in the middle. And oxygen reacts with the iron ion. To describe what's happening there mathematically, we can look at this saturation curve. Okay, the partial pressure of oxygen's on the bottom, and the percent of oxygen that's attached to the hemoglobin is on the y-axis, what we call the saturation. Okay. In the lungs, the partial pressure of oxygen is around 100 millimeters mercury. It's actually a little bit higher than that, but that's essentially the driving force. When partial pressure of oxygen is at 100 millimeters of mercury, hemoglobin is going to be fully saturated with oxygen, okay? Which means in the lungs, the hemoglobin preferentially binds to the oxygen, okay? Gives up the carbon dioxide and the hydrogen ions and picks up the oxygen. As the blood moves from lungs out into the tissues, the partial pressure of oxygen drops to about 40 millimeters of mercury. And at that point, hemoglobin is just a little bit above 70% saturation, I guess that is. So here's 60 and here's 80, so this line here is 70. And so the intersection of 40 millimeters of mercury and the saturation curve is right here, which has got to be 73, 74%, somewhere in there. Okay. Now, your blood is going to be near 100% 100 millimeters of mercury worth of oxygen in the lungs because it's picking up all of that oxygen. But as it moves out into the systemic circulation, the percentage or the partial pressure of oxygen in the blood is going to drop. It can get as low as 40, but it's going to stay more in these sort of middle ranges because there's going to be a mixture of how much blood there is depending on which blood's going to capillaries in different regions. So usually the saturation of hemoglobin is going to be somewhere up here in the around 90% range. Okay. If you've ever been to the hospital and they've put a little clip on your finger, they're measuring hemoglobin saturation. That little clip they've put on your finger is probably a pulse oximeter, which does two things. One, it measures your pulse, which it can do based on the other measurements it's taking. And oximeter is referring to the fact that it's measuring the amount of oxygen in your blood. The way it does that is it just looks at the color of your blood. Okay? The reaction of oxygen to hemoglobin makes this reddish color, and when it releases the oxygen, the hemoglobin loses that reddish color. So by shining a light into the blood and measuring the reflection off of the blood, the reflection will change color based on the oxygen saturation in the blood. Okay. As the blood's pumping through your system, it's also going to, the volume of blood passing through the blood vessels will change. And so that's where the pulse comes from. By seeing the change in the amount of reflectivity, I think that's how it's said, in the pulse oximeter, the device can figure out when the heartbeat is sending out the highest pressure and when it's at its lowest pressure. But then also it can look at the color of that reflection, and that'll give it a clue as to how saturated the hemoglobin is. And so normal blood oxygenation levels measured at rest are going to be above 90%, probably pushing 95%. When you start to exert yourself, then you're going to start sending more and more oxygen to your skeletal muscles, and blood oxygenation is going to go down. It shouldn't really go down below this roughly 74, 75 percent saturation there, because that's about where the lowest saturation of oxygen might be. Now, if your blood oxygen levels drop even further, then yes, there can be a pretty significant drop in saturation. But that shouldn't happen under normal circumstances. When blood saturation, oxygen saturation drops significantly, then there must be some serious problem underlying all of that. So the picture I just pulled this off of here is followed by a couple of other figures that are just expanding on what's happening with the saturation curve. So oxygen saturation is actually sensitive to pH of the blood. The red line in the middle, which is a pH of about 7.4, that's pretty much the saturation curve I was just showing you. That's normal. Blood pH should be 7.4, 7.35, somewhere in there. Okay. So that red line is this saturation curve we were just talking about here. If blood pH drops to be slightly acidic, even just down to 7.2, then oxygen saturation is compromised. Okay, the teal, whatever color line that is now below the red line, showing that it takes more oxygen to saturate the hemoglobin, and it's probably never going to quite get all the way up to the 100% level. Okay, so when somebody's blood is acidic, when they have acidosis, one problem with that is it compromises the saturation of oxygen in hemoglobin. And then the opposite is true if blood is slightly basic, somebody's alkalotic, then the saturation curve is a little bit better. Okay. It might seem like, well, wouldn't it be better than to have slightly acidic blood all the time? But there's going to be other problems with that. So the pH 7.4 is kind of the happy medium, keeping everything in check, more than just the saturation. But this is a result of pH changes on saturation. The other one is looking at the effect of temperature. Okay. So some weird little printing error. This thing looks like a italic F is supposed to be a degree sign. So 10 degrees Celsius, 20 degrees Celsius, 38 degrees Celsius, 43 degrees Celsius. Just showing that as our internal temperature gets colder and colder, which has plenty of problems on its own, it actually improves blood saturation of hemoglobin. And as we get hot, blood saturation of hemoglobin goes down. Okay. So there is a tie into temperature also. So basically these three figures here are just showing the basic saturation curve and then a couple of things that have an effect on that. The temperature isn't something that I'm terribly concerned about. We're going to think a little bit more about what pH means for all of this. So I'm just pointing this out. I'm not going to talk about it because I don't really deal with developmental issues in this course, but it does mention hemoglobin in the fetus. The 4 subunits of hemoglobin that we saw up here, right? Yeah, these are 4 separate proteins. In the adult, in normal situation for the adult, 2 of them are called beta globin, so it's 2 copies of the beta globin protein, and 2 of them are alpha, I think it's alpha globin, so 2 copies of that. In the fetus, there's a different hemoglobin structure. Instead of alpha and beta subunits, it includes a gamma subunit, which is sometimes referred to as fetal hemoglobin. You wouldn't see that in an adult, but in the fetus it is present. And what that does is it alters the saturation curve for hemoglobin. Okay. So fetal hemoglobin saturation is different from adult hemoglobin saturation, which is necessary because the fetus isn't breathing directly. It's getting its oxygen through the mother's breathing and the mother's blood supply. Okay. So that's when we're talking about transport of oxygen in the blood, we're really talking about the hemoglobin thing. The saturation curves are the most important part. And then if we look at carbon dioxide transport in the blood, it's mentioning sort of the 3 different ways that carbon dioxide can be found. It can be carried in the red blood cell, meaning attached to hemoglobin. It can be part of the bicarbonate ion, which means that it's from the carbonic acid reaction, or it can be dissolved directly in the plasma. That's basically what I said here. Okay, so carbonic acid, or bicarbonate. The CO2, up here, the CO2 of carbon dioxide is going to be the C and 2 of the O's in carbonic acid, and so it's actually still the C and 2 of the O's in the bicarbonate ion. So saying that it's in the carbonic acid bicarbonate is kind of saying the same thing there. Here's where the book describes the bicarbonate reactions a little bit more, and then carbaminohemoglobin, which I already talked about. So here's where the book goes into a little bit more detail about those different things there. I guess they call this section, modifications respiratory function. Oh, no, I remember. I want to go back to an earlier section. When I was showing you this last time, I said, we're going to come back and look at regulation. Oops. Which is handled here. So controlling breathing is handled within a number of different structures in the body. The main structures are in the brainstem. So there's a medullary respiratory center, which contains a ventral and a dorsal respiratory group of neurons. And then there's a pontine respiratory group, meaning that it's in the pons of the brainstem. And then there's peripheral structures in the aorta and in the carotid artery that play a role. And then in the hypothalamus and various cortical areas in the brain, there's input, and then other sort of external or peripheral players here. This figure is sort of laying out where a lot of this stuff is. So in the brainstem, we have the medullary respiratory center, which is made up of the dorsal respiratory group and the ventral respiratory group. And then there's the pontine respiratory group, which has actually two parts called the pneumataxic and the apneustic area. The medullary part is the most important part. This is what actually controls breathing. The dorsal respiratory group, its neurons send axons out that eventually control the contraction of the diaphragm and the external intercostals. Okay? Those are the muscles that are responsible for inhalation. So these neurons cause contraction of those two. And I talk about that in lab, what happens with those lungs, with those muscles. The dorsal respiratory group can also send a signal to the ventral respiratory group and modulate or control what it's doing, which is to then control the internal intercostals and the abdominal muscles, which are right here, which are responsible for exhalation. Okay. Now, you don't actually need to control the internal intercostals and the abdominals to exhale air normally. But when you need better control of over exhalation, the ventral respiratory group goes into that. Now, the dorsal respiratory group is a unique little nucleus in the brainstem in that the neurons there are autorhythmic. Where have we heard autorhythmic before? What other system were we talking about that had cells that were autorhythmic?
The cardiac.
Right, the cardiac system in the heart. Now those were the SA node, AV node, et cetera, the cardiac conduction pathway cells. Those were cardiac muscle cells that are specialized for electrical conduction. Here we're talking about neurons that are doing the same thing. Okay. Now the way that the SA node depolarized between heartbeats is that it had this leaky system. Sodium slowly depolarize the cells until they reach threshold again. That's the same sort of thing that's happening here. There are cells in the dorsal respiratory group which are sensitive, or not sensitive, but are always leaking a little bit of sodium in, so they're always depolarizing. They depolarize slower than the SA node. The SA node needs to do it about every 0.8 of a second for a normal resting heart rate, which is actually modified by the autonomic system. But here, they're going to do it about every 5 seconds. Okay. The normal respiratory rate, basic respiratory rate, is 12 breaths per minute. So that means that there's going to be a signal from the dorsal respiratory group to the diaphragm and external intercostals every 5 seconds. Okay. They contract. You breathe in, they relax, you breathe out, and then the next signal comes along, you breathe in, you breathe out, every 5 seconds, which sets up a respiratory rate of about 12 breaths per minute. Now there's a range of breath, how fast somebody breathes, so it doesn't have to be every 5 seconds, depending on various conditions. It might be a little faster, but that's what's happening there. And the dorsal respiratory group can send signals into the ventral respiratory group if it needs some direct control over the muscles for expiration. But those are not necessary for simple breathing, what all of you are doing right now. Just the relaxation of the diaphragm and the external intercostals will allow for breathing. Now in the Pontine region, there are these two groups here, the pneumotaxic and the apneustic center, which send various inputs to the dorsal respiratory group to regulate how breathing occurs. Okay? The book goes into a little bit more explanation of what those different regions are responsible for, but they can modify DRG activity, dorsal respiratory group activity. What's not shown in this picture, but was suggested in the table I was just showing you, is that there's a number of inputs to these structures. Some of them are coming from higher centers like the hypothalamus or the cortex, and those are basically going to modify what's happening in the Pontine group. Other things are coming in from the outside, like the aortic body and the carotid body. They can send signals in to modify what's happening in the medulla directly, or there might be modifications to what's happening in the Pontine region, which will then eventually modify what's happening in the medulla. So there's a number of inputs that play a role here. Now in this section here, they're talking about the things that are being modified, they're talking about the homeostatic mechanisms that play a role here. Okay. Now, I want to go, and I just want to see this section that I started to go into, the modifications. It's just talking about that stuff. Okay. So I'll get to that in a second. But in the section that's just talking about what modifies respiratory function, the main stimulus that causes regulation of the respiratory system is actually carbon dioxide. And the partial pressure of carbon dioxide, or pH, will increase breathing. Now, because of the carbonic acid reactions that I showed you earlier, carbon dioxide and pH are actually very closely tied together. Okay. So let me just go back up to the top here where I was talking about that. So carbon dioxide is directly, for every carbon dioxide molecule that you have in your body, it can combine with water and make carbonic acid. And carbonic acid will then dissociate into hydrogen ions and bicarbonate ions. The hydrogen ions in the red blood cell are mostly going into hemoglobin, but they can move out into the surrounding tissue to some degree also, as well as the carbon dioxide that's in the plasma. It's floating around in water, and carbon dioxide and water can interact and make carbonic acid. So there's necessarily an increase in hydrogen ion concentration in the blood with carbon dioxide levels rising. Okay. The underlying pH or partial pressure of carbon dioxide is regulated and stabilized. But if you aren't exhaling as much air as possible, or as much air as you need to, carbon dioxide levels are going to increase, and therefore pH levels will increase also. Okay. And when that happens, the signal goes to the medullary respiratory groups and stimulates respiratory rate so that you breathe faster and you're exhaling more carbon dioxide over time. When you do that, you will necessarily be breathing in more oxygen, but it's secondary to what's going on. The system is really regulating carbon dioxide levels and therefore pH. I showed you earlier that there is an interaction between pH and oxygen saturation and hemoglobin. So everything's really kind of interacted together. And if your body's regulating carbon dioxide or pH, it's going to have an effect on oxygen levels too. But really your body doesn't say, oh, we don't have enough oxygen, we need to breathe deeper. It's saying we have too much carbon dioxide, we need to breathe deeper to get rid of it. When you have low oxygen levels, the response in your body actually is to make more red blood cells. Because if you don't have enough oxygen in your system, then it must be that there's not enough red blood cells carrying oxygen around. So if you make more, you'll have more capacity to carry oxygen. Which doesn't really address getting oxygen into your body, but if your oxygen levels are down, your carbon dioxide levels are probably going to be up. And so that's going to stimulate breathing. You'll get more oxygen into your system. And there will just be more red blood cells available to make it, available to carry it if need be. Okay. So in this section here, they're just talking about a few different things that can alter respiratory function. And I do want to just mention, because I've been talking about air pressure at sea level and that sort of thing, that there are some differences to consider based on altitude. So we usually just work with sea level in mind. Here, like I said earlier, we're well, on the 5th floor of this building, we're about, what do we say, 230 some odd feet above sea level. But the pressure, if I remember when we looked at it, Jessica, was like 752 or something like that.
Yeah.
Isn't necessarily that off. But if you go to Boulder or Aspen, Colorado, or Pikes Peak, and you start to get to very high elevations, so 5,000 feet is going to be essentially a mile high. And then Aspen and Pikes Peak gets even higher than that. The atmospheric pressure, the overall pressure starts to drop significantly. Okay? Like I said, on this floor here, we might be 752 or something like that. But when you're getting up to a mile or two in altitude in Colorado, then you're going to see some really significant drops there. Okay? So the partial pressure of oxygen is dependent on the total atmospheric pressure. The amount of oxygen present is going to be pretty consistent. It's going to be that 20.9%. But 20.9% of 632 is only 133 millimeters mercury. And then it drops even more as we go further up. Oxygen saturation starts to decline when partial pressure of oxygen goes below 100. Okay. Yeah.
So when someone's taking their oxygen saturation, like if they're at the doctor's office, do they have a consistent number for what the elevation would be there? Would it be the same?
Either the doctor's office has calibrated the pulse oximeter to deal with that, or well, no. It's an interesting question. Yeah, I mean, if you go to a doctor's office in Boulder, your saturation levels are going to necessarily be lower just because of where you are. So whether the doctor's office takes that into account or not doesn't really matter, because it's going to affect everybody equally that comes there. Now, people that live in Colorado, presumably, since they're dealing with less oxygen, are going to have their baseline, red blood cell count or something like that probably be higher. I don't know specific numbers on that, but I wouldn't be surprised by that. Now, at sea level, partial pressure of oxygen is 159. When we're in the lungs, partial pressure of oxygen is actually 100, 105 millimeters mercury. So the air that's in your lungs versus the air that's outside of your lungs is not the same. Okay? Actually, there's quite a bit more carbon dioxide taking up space in the air. So there's less there's less oxygen in there. So the partial pressure of oxygen inside your lungs is necessarily lower than what it is outside. So to say that we're in Boulder and outside the partial pressure of oxygen is 133, inside your lungs, it's probably already below 100 millimeters of mercury. So we're already going to see potential oxygen saturation problems at that elevation. Now, there's a point at which, as we're talking about these really high elevations on these mountains, there's a point at which oxygen pressure in the air around us just isn't enough to get oxygen into our blood. Okay, and so people need oxygen tanks when they're hiking up high mountains. I'm certainly well aware of that necessity on Mount Everest. I don't know too much about Pikes Peak or Denali in Alaska, but I'm sure that there's some point that hikers are going to need oxygen tanks to help them hike a little bit. And that's just so that the oxygen that they're breathing in is actually a richer mix, and so the partial pressure will be up high enough that they can get oxygen into their system. Now, scientifically, it should mean that people that live in Boulder are better at carrying oxygen in their blood than people elsewhere. If oxygen levels are low, your kidney will cause an increase in red blood cell production. So you would expect, if you looked at people in Boulder, their hematocrit numbers should just across the board be higher than people in New York City. I don't know if that's true, but scientifically it makes sense. Now, athletes want to maximize their oxygen use so that they can get as much oxygen to their muscles as possible. Surrounding professional biking, there's a lot of news several years ago now about doping scandals, which were mostly the athletes training under low oxygen conditions so that when they're actually riding, their system's getting more oxygen than they're used to, so they can do more work than they normally would. One way that can happen, one way that that's possible, is for the person when they're training to actually take about a pint of blood out of their system. So they're training with low blood volumes, and therefore there's going to be fewer red blood cells. And then right before they go in for the race, they get that pint of blood back. Okay? That's one of the situations that results in the doping scandals that are mentioned. I don't know quite how they can determine that has happened, but there are going to be some sort of molecular signs in a person's blood when they traditionally have low blood volume, and suddenly they increase their blood volume again real quickly. It would seem one way to do that would be to actually train at higher elevations, okay, and then perform at lower elevations. Your body should be more accustomed. I don't know how effective that is, and probably the best evidence for that is that the sports teams that you hear about winning a lot of things don't tend to be from the higher elevations in Denver, in Colorado. Okay, so I think Denver's professional basketball team might be called the Nuggets. Yeah, that's what I thought. I suddenly was thinking, like, maybe that was Nevada, but yeah. So is it Denver Nuggets? Yeah, you don't hear them necessarily winning against some of the bigger teams. You would think that because they train at a higher elevation, if they go play in in Los Angeles, which is going to be sea level, that they're going to be getting a lot more oxygen to their muscles, and they should be able to perform much better than their opponents. But I don't know that there's any real evidence of that. So, and I don't really follow professional basketball that much, so I can't quote statistics, but I'm pretty sure that that's not really the case. So the difference here is probably not that significant to make athletic training better at higher elevations, but it would be one way to do that. And people that do hike higher elevations are going to train for it. They're going to get their body accustomed to the low oxygen levels as much as possible, but they're probably going to need to supplement it somewhat as they get closer to the summit and the oxygen levels really drop off there. Okay. Which is some of what's talked about in the acclimatization. I didn't pronounce that wrong. The next section here, talking about other aspects of that. So, then it goes on to embryonic development in the respiratory system. Like I said before, I don't really deal with developmental issues in this class too much. If you're interested, you can look at it. There are some differences in fetal respiratory function versus adult respiratory function. If you're interested in looking at that, but there won't be questions on that in the part 3 or part 4 assignments. We're going to concentrate mostly on the anatomy, breathing, gas exchange, transport kind of things. That went over there. Any questions about the respiratory system? So that actually wraps up what I wanted to cover about the respiratory system. I could move on and start talking about the digestive system and start that up, but I don't think 10 minutes is enough time to really get into what I want to there. So we'll just call it a day here, and today is Tuesday, right? Yeah. So Thursday we'll start the digestive system. Oh, no, never mind. That's for my other class. Have a good day.
When I get down to the bottom where I can take a screenshot of what the readings